Good afternoon, children. So, in the last class, we were completed the syllabus until the dihybrid cross experimental, isn't it? Today we will continue the further important topic, that is nothing but non-Mendelian inheritance. See here, in general you can ask me, sir, what is the non-Mendelian inheritance? Until then whatever we were discussed, the laws, this are all it is applicable, isn't it? It is applicable to the Mendelian experiments or Mendelian law. So that's why those what we call Mendelian laws of inheritance. But there are few scientists. Okay. So as I said in the beginning only, there are three names of the scientists, isn't it? So those scientists also, that means by namely in 1900 the Mendelian principles were rediscovered by, there are three names of the scientists, right? So that is nothing but Hugo de Vries from the Holland, Karl Correns from the Germany, and it is Deschmark from Austria, isn't it? So those, the scientists, they continued the experimentations, whatever Mendel it is given by based on the pea plants, they try to cross check by considering the different species, so that they will come to know that these whatever the laws given by the Mendel, it is not applicable to further species. So just like the plant, a snapdragon or Anthurium species is common as a dog clover and 4 o'clock plant. Okay, so then those will be whatever the laws they discovered by considering the different plants. So then they call them as a non-Mendelian inheritance. So they are also known as popularly the deviations from Mendelian principle. Okay, so today we will discuss all those things. What are those will be? See, in non-Mendelian inheritance, so these are all the 5 important topics, or the terminologies we will discuss as well as the topics. In that one, the non-Mendelian inheritance, first one is incomplete dominance. Okay, then we will discuss about the codominance. Later we will discuss about the multiple allelism, and after polygenic inheritance, and also pleiotropy. These the 5 important topics. It is very important for the purpose of the competitive as well as your theory also. Okay, so let us study one by one. Fine? Yeah. In that one, first one is incomplete dominance. Okay. So remember the incomplete dominance, it is also known as partial dominance or blending inheritance. So how can we define what is the incomplete dominance? It is the inheritance in which the offspring shows intermediate characters between two the parental characteristics. See, what is the meaning of this? It is the inheritance in which offspring shows intermediate character. See, underline the terminology intermediate character. What is the meaning of intermediate? According to the law of Mendel, if we crosses between two parents by considering any character, for example, when we conducted the experiment between the tall and dwarf, what we got? We will get in F1 generation in intermediate character, what we got? We got the dominant character, isn't it? That is nothing but in F1 generation all plants become heterozygous tall. But the thing is, in this case, in case of the flower snapdragon, it is common as a dog flower or Anthurium species, and Mirabilis jalapa, it is popularly known as 4 o'clock plant. In this plant, whenever they conducted the same monohybrid cross, what happens? Either they are not resemble the dominant character, and they are not resembles the recessive character. Here the cross between the homozygous red and white, it produced pink flower plants. So that means intermediate character between the two parental characteristics. Okay, so here the diagram, the photography also given. The snapdragon flowers, generally it is used in the puja purposes and all. Okay, isn't it? As well as the decoration purposes. And 4 o'clock plant, it is commonly grow as a shrub, or we can also call it a weed. So these are all the different type of the flowers we can identify. In this case, that is Mirabilis jalapa. Okay, and actually I was telling about why it will be considered as a non-Mendelian inheritance. See, when we conducted the monohybrid cross, it is given by the Mendel. After the experimentation, for example, if you take here only, that means the considering with respect to the parental generation, when we crosses between the dominant reds, so that is nothing but homozygous red flower plants, by considering the flower petal. Okay, yeah, with homozygous white flower plants, what should we get? According to Mendel, heterozygous gamete, that F1 generation should be, according to Mendel, I am telling you, according to Mendel, it should come heterozygous red. But here in this case, I mean the snapdragon. Okay, snapdragon are Anthurium species, as well as the 4 o'clock plant, they got pink flower, heterozygous pink. Okay, so then they amused. So what is happens here? Okay, what is happened? So whenever they conducted the experiment in F1 generation, okay, the phenotypic range, whatever the generation appear, the pink was not resembling to any one of their parents. When they continued the similar experiment, okay, in F2 generation, by that means the selfing with respect to the F1 parents, they got, see here in the opponent square you can identify, that means capital R 1, there is a red, and capital R small r pink 2, and small r white 1. So that they can come to know a phenotypic ratio, they got 1:2:1. That means red is 1, the pink is 2, and white is 1. Okay. And what about the genotypic ratio? Yes, the genetic constant also similar. That capital R is 1, capital R small r is 2, and small r is 1. Okay. So that means thus the phenotypic and genotypic ratios are the same in case of incomplete dominance. Okay. But what about the Mendelian inheritance? In monohybrid cross, we got 3:1 as a phenotypic ratio and 1:2:1 as a genotypic ratio. Isn't it? Do you remember? Okay? Yes. But the thing is, here we got, that means the phenotypic and genotypic ratios are same. There is no difference between the phenotypic and genotypic ratio in this species. So this means that R was not completely dominant over R, that is small r. What is this R? R nothing but the flower petal color representing gene. Okay, so it is not completely dominant over small r. So that what happened? The partially, or we can also say the blending of inheritance happened. So that's why it is popularly known as incomplete dominance. Okay, so this question they can ask repeatedly in the competitive also. And remember, actually whatever the Mendel, he conducted the hybridization experiments, he got results also. He considered only 7 pair of contrasting characters. Isn't it? Do you remember? There is the characters, the stem height, seed shape, seed color, pot shape, pot color, flower position and flower color. But pea plants also show incomplete dominance in other traits. For like for example, traits of the flower, the serration of the flowers, even sometimes the serration considering with respect to the leaves also. So they shows the pea plants also show incomplete dominance in other traits. Okay, but Mendel is not considered those, and he is not conducted the hybridization experiments on the not accept these 7. Okay, but remember these pea plants also show incomplete dominance in other traits except those 7 characters. Okay, yes. And after this one, let us study, there is another important topic, that is codominance. Okay, incomplete dominance is clear, right? Okay, I think it's clear. Fine. Yeah. So after this one, there is another important topic which is also comes under the deviations from the Mendelian principle, that is codominance. The terminology itself indicates. Okay, what is the meaning of this one? Co means both. Okay, it is the inheritance in which both alleles of a gene are expressed in a hybrid. Okay, so we can also explain in another manner. So that means what is the codominance? When the alleles for 2 contrasting characters are together in heterozygous condition, both the alleles express their character together is called codominance. Okay, but remember AB blood group in human as well as coat color in rabbits. See, in general we can also give example ABO blood group, but remember out of this ABO, AB blood group is exact answer for codominance. I will tell you why. Okay, I will tell you why one by one. Okay, and if you consider in case of the animals also, the coat color in rabbit is also best example for the codominance. See here in the right side, just observe the whatever it is given the antigens shares. Generally the ABO blood groups are controlled by gene I. Do you remember when I was telling about the blood groups? Okay, I told no the last year also. I hope you can remember the Carl Landsteiner in 1900 is the first person who classified the human blood into the 4 groups such as A, B, AB and O, isn't it? And the blood groups of the man is determined by 2 kinds of proteins in the blood such as antigens and antibodies. Okay. Again you can ask me, sir, what is the antigen and what is the antibody? Okay, note down, antigens are also known as agglutinogens. Okay, what is agglutinogens? Again you can ask me. The agglutinogens or antigens are the glycoproteins. They are nothing but the polymer of sugar present on the membrane of RBC, that is the red blood corpuscles. Here the diagrammatic in clear picture is given, and there are 2 types of the antigens such as antigen A and antigen B. We can identify in the blood groups. Okay, then you can ask me, sir, what about the antibody? Agglutinins. These are also called, antibodies, they are nothing but agglutinins. Okay. Agglutinins, these are the proteins present in the blood plasma. Okay, antigens, where we can identify these antigens are the glycoproteins, or we can also say the polymers of sugar present on the membrane of RBC. But what about the antibody? These are the proteins present in the blood plasma. Okay, so there are 2 types of the antibodies present in the blood plasma such as antibody A and antibody B. Okay, you should remember about this one, then only you can come to know how the blood groups and all can be classified. As I said, the ABO blood groups are controlled by the gene I. You can ask me again, sir, what is this I? I is nothing but it refers to the isohemoagglutinogen gene. Okay, which exists in the 3 allelic forms. Okay, which is allele means which is exists in the 3 allelic forms. I to the power of A, I to the power of B, and commonly I or we can small I. Okay, so IA and IB are the dominant over I generally. Okay, see here, IA and IB produces a slightly different form of the sugars compared to the one another. Okay, but what about the small I? Allele small I does not produce any sugar. Okay, you should identify clearly the antigen positions or the shapes which is present in the blood type A, antigen in case of the B, and antigen in case of the AB blood group. Okay, what about the O? Neither antigen A nor B. Okay, then those will be considered as the O blood group. Did you got it? Yes. If you understood this one, then only you can identify and you can understand the further things. Okay? Yes. So here the genotype of the different blood groups. So I told you last year also, earlier we had a blood test in our syllabus, but in new syllabus it is removed. But generally if you go to the doctors or if you go for the serological test, first they will check the blood groups. Then after they will conduct the further whatever the serological test, okay, for the diagnosis purpose of the different diseases. But here let me to tell you briefly, the blood sample they will collect from the patient or the person. Okay, then what they will do actually, they will collect the blood samples on the slides and they will add or they will put a less amount of the, that is that is nothing but for the purpose of agglutinations. Okay, for the purpose of the agglutination, they will put a little bit of the antisterals. Okay, if they find the agglutination is nothing but the clumpy nature. You can identify clearly in the anti-A. If the clumpy thing or the powdery like appears, if it happens in A, then they will say that the blood type is A. Okay. If the same thing it happens, that means the agglutination if it happens in the B spottings, then they will consider the blood type is B. Sometimes agglutination happens in the both the spottings, then they will consider it the AB. Okay. If it happens on both, then it will be AB. If there will be no agglutination on the both the spottings, then they will be considered O blood groups. So this is how exactly, okay, they will conduct in the blood groups also. Okay, so the genotypes of the different blood groups. Remember, the blood group A, it is found in case of both homozygous condition as well as heterozygous. Homozygous means I to the power of A, I to the power of A, then it will be homozygous. Heterozygous means I to the power of A and small I. Like that, a similar thing we can identify in case of the B blood group also, homozygous and heterozygous. But what about the AB? Both will be present. Okay, both will be present. It is I to the power of A and I to the power of B. And what about in case of the O? There is no. That's what I said. What is this? Okay. See, in case of the O blood group, antigens are nil, but antibodies are present. Remember, the antigens are nil in case of the O blood group. Antibodies will be present. Okay, so this is how exactly the genotypes of the different blood groups we can identify. And I was telling about, see, this table is useful, okay, for the prediction of the blood groups. So this table they can ask in examination also. If you remember how to write this one, on the basis of this one, they can ask the questions in competitive also as well as theory also. Recently, whatever your seniors they have wrote the examination in the mass, one question, in one mass question they were asked. Okay, so let me to tell you in the easiest manner. So this table is the indication of the blood groups. How it will be considered the alleles from first one, and in the first table it's the indication of the parent one. Okay. Just imagine, then alleles from the parent 2. Okay, or in another manner, I will tell you, for example, the parent. Okay, parent 1 I will consider as a father, then parent 2 I will consider as a mother. If the parent 1 marries with parent 2, What will be the genotype of the offspring in a homozygous condition? IA. Then what will be the blood type? Surely the phenotype of the blood group is A. Are you getting? Okay. Next, what about the heterozygous condition? If the parent 1 is I to the power of A, if the parent 2 small I, that means if he or she marries with the blood group O, then what will be the genotype? I to the power of A and small I. But in heterozygous condition, A is dominant over, that is small I, then the blood group again the phenotype is A. What about the B? Yes, if the parent 1 and parent 2 both are having the blood groups B, the genotype of the offspring B and B. This is a homozygous condition. Again the blood type surely will be the B. Then heterozygous, I to the power of B and small I. The genotype of the offspring heterozygous, I to the power of B and I. Again the blood type will be B. This is a heterozygous condition. Now comes to the exactly what I said. There is a codominance. Yes. Either the parent 1 is having A blood group marries with the B, or if there will be another chances, the B marries with A. In both the cases, heterozygous, the genotype will be AB, so that the blood type will be AB. So in these both cases, we can say yes, when A, that is capital I to the power of A and I to the power of B are present together, they both express their own type of the sugars. This is due to codominance. Okay, this is due to codominance. But what about in case of the I? If both the parents, see generally you must have heard about, okay, the blood group O, that too specially O negative blood group is very rare. Do you heard about the O negative? Yes, the O negative blood group is called universal donor, and AB positive blood group is called universal recipient. Okay, if the O blood group person marries with a similar group, surely that blood type will be O. Okay, yes. So again, you can ask me, sir, how the positive and negatives they can identify. Again, it is based on, there is another anti-serum they will add. Based on that one, they will identify either it is A positive or A negative, like the AB positive or AB negative. Surely like that only O positive or O negative. Okay, yes. So this is how generally so they will identify the blood group also. Okay, yeah, I think it's clear. Yes. So here the example, okay, because generally in exam also they will ask identify the blood group of the children by using the following blood groups of the parents, like they will ask the question. So that in such cases you should work out like a checkerboard, so that you can easily write down in exams as well as without confusion in your competitive also. I will consider the father is having heterozygous A blood group and the mother, she is having heterozygous B blood group. When they cross between each other, see what we will get? I to the power of A, I to the power of B, then it will be blood group will be AB. If I is crosses with I to the power of B, then heterozygous B. If the I is crosses with I to the power of A, then the heterozygous A. If I is crosses with I only, you can ask me, sir, why we are written off of? You know very well that during the formation of gametes what will happen? Only half of the chromosomal number, it will move towards the further generation, isn't it? Again after the fusion they will become the, so and again it will become twin. Okay, so in this case, what are the possibilities? Yes, AB blood group 1 A heterozygous, B heterozygous, and O blood groups. These are all the possibilities. Offspring are formed A group heterozygous, B group heterozygous, AB group, and the last one is O group. Okay? Yes. There is another case. If the father is having a blood group, AB blood group, if they crosses with mother, that means O blood group, then what are the possibilities? What are the possibilities? The offspring are A group, that is heterozygous A, yes, then again the B group, again heterozygous. Okay, so this is how exactly to identify the blood group of the children by using the following blood groups of the parents. If you learn this one, so in examination, I told you, they can ask this type of the questions in theory as well as in practical also. Okay, so especially in your competitive, you can write this one. Okay, yes. The next topic is multiple allelism. Okay. So what is this multiple allelism? The definition, first of all, see, there are a number of cases the students they get confused with codominance as well as the multiple allelism examples. I told you in case of the codominance, AB blood group in man, coat color in rabbit means codominance. ABO blood grouping means it is multiple allelism. Okay, remember that's why I am telling you in the beginning only. Okay, yes. What is multiple allelism? The definition itself indicates it is the presence of more than two alleles. It is the presence of more than two alleles of a gene to govern same character. That's what I said. The alleles of a ABO blood group, there are three alleles which is present. Okay, that is, these three combinations, they are responsible for the blood groups, isn't it? There is the presence of more than two alleles of a gene to govern the same character. That means these more than two alleles, they governs the particular, that means they controls or they decides the particular blood groups in humans. Okay, so in an individual only two alleles are present, but generally during the formation of gametes, whenever that 2n is get converted to n, so in an individual only two alleles are present. Okay, but multiple alleles can be found only in population, not in a particular individual person. Remember, in an individual only two alleles are present, but multiple alleles can be found in only in a group of population, or we can also see other populations. Okay, here the example also given. Okay, see, let me tell you once again. If there is a definition, you can write it is the presence of more than two alleles of a gene to govern the same character, or you can also write in another form, more than two alternative form of a single gene occupying the same locus on the homologous chromosomes. Okay, and control a single character is also called multiple alleles. Okay, is it clear? The phenomena is called multiple allelism. The alleles are known as multiple alleles. Okay, yes. So this is another one, the polygenic inheritance. See, you can ask me, sir, what is poly? Poly means many. Okay, so generally when it comes to the human skin color as well as the human height, let me tell you, in generally, the skin color generally sometimes, if the whatever the parents, if you just observe, sometimes the fair person marries the dark person, sometimes the medium color, but sometimes if they compare the color of the human skin with respect to the parents to progeny, it will be different. Similarly, in case of the human height also, There is no guarantee that if the father is tall, the children should also become tall. There is no guarantee. Sometimes the children, even if the dwarf parents, okay, so they will give birth to their, later days, they may also become the tall. So this is how it will be actually, okay. So this all it is because of this polygenic inheritance. Remember, what is the polygenic inheritance? It is an inheritance in which some traits are controlled by several genes. Okay, what is the polygenic inheritance? It is an inheritance. Inheritance is the phenomenon. I told you in the beginning also. It is the inheritance in which some traits are controlled by several genes. Several genes are also known as multiple genes. Okay, where we can identify the best example, as I said, human skin color, human height. Okay, it considers the influence of the environment. Why it will be? Because it considers the influence of the environment. Okay, in a polygenic trait, the phenotype reflects the contribution of the each allele. Thus, the effect of the each allele is additive. Okay, here only, so they have given. So if you consider the white or very fair, or then we can also give the tint. Okay, example. So we can also give the hues, a shade, and blacks. Okay, so based on the parents, and in case of the offsprings, we can identify the different type of the human skin colors. Like that, in case of the human height also. Okay. A tall, a dwarf, or medium, or we can also say intermediate. So these are all the different type of the possibilities. Okay, these are all the possibilities. It is because of the polygenic inheritance. Okay, yes. So here the example, actually the human skin color, the workout, it is not present. Okay, the human skin color, this is actually for the purpose of understanding as well as identification. So this is useful for your need. Okay, note down this one clearly. Consider in case of human skin color, assume that there are three genes, capital A, capital B, capital C. They are the dominant controls the human skin color. These are the three genes which controls that is human skin color. Okay, the dominant genes, so generally dominant forms the capital A, capital B, capital C, responsible for the dark skin color. In case of the humans, dark skin color is the dominant. Okay, what about the recessive? Yes, small a, small b, small c for the light skin color or fair color is considered the recessive forms. Okay. When they cross between each other, what are the possibilities? The genotype with all the dominant alleles. Okay, when they comes in contact of each other, if the both the cases, if the dominant alleles comes in contact of each other, surely it will give darkest skin color. Okay, you can ask me sometimes, sir, why the particular region peoples of the different countries, why they are extremely or darkest skin color? Because of this condition. Okay, when the genotype with all the dominant alleles, so then that will be, it gives rise to the darkest skin color. Then what about the lightest? The genotype with all the recessive alleles. Okay, if you work out, just write them as it is. Okay, you can work out. Okay, made as a gametes. Capital A, capital A, capital B, capital B, capital C, capital C. At the same time, you can write down small a, small a, small b, small b, small c, small c. Make a gametes. Then you will get a capital A, capital B, capital C as a gametes. Then small a, small b, small c as a gametes. Cross between each other. Then you can get the intermediate also, isn't it? Capital A, small b, capital B, small b, capital C, small c. Then it will be considered as the intermediate. The genotype with 3 dominant plus 3 recessive alleles gives the intermediate skin color. Okay. So thus the number of each type of alleles in the genotype determines the darkness or lightness of the skin. Okay. Yeah. The another one, the last topic, which is comes under the Mendelians. So that means non-Mendelian inheritance or the deviations of the Mendelian law, pleiotropy. Okay, what is this pleiotropy? So here, in case of the pleiotropy, a single gene exhibits multiple phenotypic expressions. Such a gene is called pleiotrophic gene. Okay, I will repeat once again. In case of the pleiotropy, a single gene exhibits multiple phenotypic expressions, multiple physiological expressions. Such a gene is called pleiotrophic gene. The phenomena is called pleiotropy. That gene is called pleiotrophic gene. Okay, so in most cases, the mechanism of pleiotropy is the effect of a gene on metabolic pathways, which contributes towards the different phenotypes. Okay, so these are some of the examples: the starch synthesis in pea plant, phenylketonuria, sickle cell anemia. Okay, so here the diagrammatic representation also. Consider a single gene which is present in a chromosome. It may appear as a stride A, or it may appear as stride B, or it may appear as stride C also. Okay, there are three possibilities, isn't it? Then, in phenylketonuria and sickle cell anemia, the mutant gene has many phenotypic effects. Example, phenylketonuria causes mental retardation, reduction in hair and skin pigmentation. About these all, the disorders and all, in this chapter only, last topic. Okay, we will discuss again, in detail. Here we will discuss about only the examples. Okay. The next one, see here the case. In case of the humans, the phenylketonuria and all, we can give examples, right? But in case of the plants, the pleiotropy is possible. Okay. If you take an example of the starch synthesis in pea plants, the starch is, that is carbohydrate, is synthesized effectively by capital B. Okay, that is both the dominant genes. Therefore, large starch grains are produced in case of the pea plants. Small b have a lesser efficiency in starch synthesis and produces smaller starch grains in case of pea plants. Okay, but when the starch grain size also shows incomplete dominance, that means intermediate size seeds. That means when we cross, or if they undergo cross between effective starch synthesis, that is capital B, gets a large size seeds with a lesser starch synthesis, small b, small size seeds, the gametes will be like this, and finally it leads to the formation of incomplete dominance, that is capital B, small b. Intermediate size seeds also produces. So this is the best example, that is the pleiotropy, okay, in case of the plants. Okay, I hope you can identify. So today we were discussed about what are the things. Yes, we were discussed about the 5 important topics, that is in the deviations of the Mendelian law: incomplete dominance, codominance, multiple allelism, and also polygenic inheritance, as well as the pleiotropy. Okay, so let it be. Next class we will continue the remaining things. Okay, thank you. Have a nice day.